So yeah, super excited to give this talk about an end-to-end AgTech use case that I experimented with in FiftyOne as part of giving my talk. So super quickly about me, my name is Prerna. My background is in machine learning and specifically in computer vision for defect detection. And now I work at Voxel 51 in the customer success team, where I get to work with our enterprise customers, small and large, on a bunch of different problems in the industry, AgTech definitely being one of the primary ones of them. And I'm super excited to chat with you all today. Let me go to the next slide here. Yeah, so today's agenda is going to be the first little bit of this presentation will be me talking about the problem statement and the AgTech use case I picked to work on. And then as part of this AgTech use case, I'll talk about the data exploration and curation steps that I went through. And as any good machine learning problem usually involves, I went on a side quest with Dino V3 as part of my data exploration and curation. So I'll take you along on this journey as I talk about how I learned more about Dino V3 as a part of exploring and curating my data. After that, I did some training and evaluation and failure mode analysis. So I'll talk a little bit about what was involved in that process as well. And then at the end, we'll tie a quick bow on it with my key takeaways from this whole journey. Yes, also, if you have any questions throughout this whole presentation, please feel free to put them in chat, in the Q&A section, and then I'll answer them at the end of the talk. Okay, awesome. So as I was looking at what use case I wanted to work on, I was looking at various open source AgTech datasets that are available. And for people who get the heebie-jeebies, I apologize. This pest detection dataset is interesting, but can also give you the heebie-jeebies because of all of the little insects we're going to look at. But this was a pretty cool dataset that I came across. It was a large-scale benchmark dataset. I think this was released in 2019 at ICCV. Since this is an AgTech-specific use case or an agriculture-specific use case, I thought it would be really cool to work with this dataset. A little bit more about this dataset. I think there are a total of 78,000 images that are gathered, and then 18 to 19,000 of those have object detection labels on them. So that 19,000 is the core set that I will be working with. And my goal with this whole process was to build a quick solution for a real-life agriculture problem and identify a good starting point at the end of the day from which we can iterate from. And that is basically what you'll see in the takeaways as well. Quick little note about this dataset also. There's about 102 classes in this dataset. And I will say there is like a hierarchical taxonomy in these classes, meaning that many of these 102 classes fall under a parent class. But that information wasn't as easily available as the breakout information of this breakout into these lower level classes. So I've just stuck with the 102 classes. But right from the get-go, that was something that I identified as being an issue with my approach, which is the lack of having these lower classes roll up into a parent class, because you'll see a lot of overlap between these 102 classes. Okay, awesome. Yeah. So once I identified a dataset, I loaded that dataset into FiftyOne and then began my exploration and curation process. So in this talk, I'll kind of quickly jump between my presentation slides and also just exploring the data in FiftyOne. But as a part of my dataset exploration and curation, I looked at a bunch of data quality metrics. I looked at the distribution of the classes to see how balanced those classes are, and also explored some labels. So let's go into the dataset here. So this is the basic dataset that we're working with. You'll see, because of the order in which it was loaded in, all of the images of one specific class are available at the top. But let's say I wanted to get a quick overview of a mixture of different samples. I can really quickly shuffle the data in here and see a shuffled subset view of the images that are returned. So we'll give this a second to load, but you will basically see, and we can zoom in here a little bit as well. But you'll basically see a bunch of mixture of classes of the 102 classes. And you can also see that there are classification labels as well as object detection labels on top of this. One quick note here. The authors only provided object detection labels, but something that I noticed was usually in one image there was only one type of insect or pest. So what I did was I extracted the label or object detection label and added it as a classification label. So we could quickly kind of see in this grid view what types of classes are present. So the next thing that I wanted to do was just understand the quality of my data. So I went into this data quality panel, and then we have a bunch of kind of standard data quality metrics that are used across the computer vision industry. But these do use traditional computer vision metrics. So we have brightness, bloodiness, aspect ratio, and so on. But the specific ones I was interested in were bloodiness as well as near duplicates. So as I was scrolling through the data, one thing I noticed was there were a lot of duplicate copies of the same image over and over again. And a really great way to identify what those duplicates might be is using this near duplicates metric. What this does is looks in the embedding space and finds the cosine distance between every image and every other image in your dataset. And that will help you, and then you're able to put that cosine distance on a scale and between an image and its nearest neighbor. And that's essentially how you're able to find the nearest duplicates metric. And yeah, basically looking at the near duplicates here, right? So everything below, let's see, 0.15 cosine distance is what we flagged as possibly duplicates of each other. And you'll see that that is kind of accurate. So we have a bunch of images where they look basically like exact copies of each other. There are also other images which are the same pest, but from a slightly different view. Like, for example, these two look like the same pest from a slightly different view, whereas these two look like exact duplicates. So it's a very easy way for me to understand if there are problems in my data set, because at the end of the day, I want to make sure that my test set is completely independent of my training set and there's no data leak between the two. I will say after doing this, I did not use this metric to further split up my train, val, and test data sets for lack of data leakage, because I wanted to keep my experiment very constrained, and everything I was able to achieve that I'm going to show you in today's talk was something I was able to do in a matter of 3 or 4 hours. So I did want to constrain my efforts on this to see how far I can get in those couple of hours. But another note that I will say I could have done for improved performance and for more robust models is making sure that that I used this near duplicates metric to ensure there was no data leakage between train val and then the test sets. Okay. The other thing that I spoke about was looking at our distribution of classes. So let's go into our histogram of class distributions here. So all this is doing is looking at the distribution of the available classes in my main data set. And you can kind of see that it is definitely an imbalanced data set, right? There are a few pests that are overrepresented in my data set, and then the number of instances of the other pests starts kind of starkly dropping off. Another pretty obvious note that you can note or takeaway you can take from this plot is that you are going to have imbalanced performance because of this imbalanced data set. So it's likely the overrepresented classes will have better performance and the underrepresented classes will have poorer performance. And that is also something that we can see when we look at our model evaluations. So a little bit of foreshadowing here, but something to keep in mind what this plot looks like as we go through the rest of this process together. All right, and then the last thing that I was doing was exploring our labels. So that's something as simple as just going into my pest class here and then filtering by various labels to just see what the different labels look like. So let's look at aphids here. These aphids are a pest that I've personally dealt with because with my outdoor plants, I've seen aphids grow pretty extensively on, I think it was my tomato plant specifically. So yeah, definitely a pest I've personally dealt with. And just looking at what these samples look like and getting a quick overview as you filter by your labels is very convenient here. So that was another part of the data exploration process that I did, which is just take a look at what the different classes are, get a sense for what my data set looks like. Alrighty, let's keep going. Here, I wanted to talk about my side quest with Dino V3. So as I was exploring my dataset more, another really powerful tool that FiftyOne offers is the ability to look at your embeddings. What embeddings enables you to do, and this embeddings visualization enables you to do, is get a sense for how your images look like in a slightly more quantitative sense than just getting a high level qualitative idea of your samples, right? So obviously I can look through my data in the grid and scroll through it, but having all of the data laid out in a 2D feature map space, so I can see the relationships between the different images, and so that I can get a very high level understanding of my data, I found this to be really helpful as well. So here, I'm looking at just my CLIP model, which is a contrastive learning model that OpenAI released a couple of years ago. And something interesting you can do is color by your pest classes. So ideally, if the model that you're using to extract your embeddings is really good at telling apart features in your specific domain, what you would see is the colors are very distinct and not mixed together. So every point I see here is colored by its class label, but I see a lot of mixing in, right? So I see this orange class, which we can start to lasso and see what this class is. This class is pretty distinct and far away from the rest of the clusters. So this looks like, yeah, it looks like it's a Lycorma type of pest. And you see also this cluster over here, green, which is kind of distinctly separate from the rest. But you start to see a lot of mixing in here, and you see more of a giant central blob. So I was trying to think about what model I might be able to use, which might be able to tell apart my classes a bit better. And CLIP was released a couple of years ago. I did want to work with a more state-of-the-art model. So what I looked at instead was the Dino model, so specifically the Dino V3. The Dino V3 paper is really interesting. And first of all, just without even uncolored, right? You can kind of see that the clusters are much more farther apart, and there's no central big blob that we see. There is some mixture or larger blobs here, but in general, it looks like the Dino model is able to put images of a specific class farther apart and is able to better tell apart features for this specific use case. So let's color by pest class label again here. And you definitely see less mixing, right? There are more specific clusters here as well. There is obviously some mixing here. And my guess is going to be the classes that mix together are actually all, lower level classes that belong to the same higher level taxonomy. Or at least they look kind of similar, which, yeah, it does look like they all look kind of similar here. For example, this is labeled as an alfalfa plant bug, but to me, and personally having dealt with aphids, it does look like an aphid also. So you can see it does kind of look like this aphid here. So it looks like these lower level classes do roll up to a higher level taxonomy, higher level class in the taxonomy. So anyway, having looked at this, the performance of Dino V3 here, I got a bit curious about the changes that Dino V3 introduces. And I quickly wanted to highlight the Dino V3 paper also here, which I found really cool and interesting. The way that they got Dino V3 to be really good at learning localized features was by something that they called gram anchoring objective, which they introduced in this paper. Essentially, what they noticed was as they kept training for extended epochs, global metrics and benchmarks got better, but localized dense task performance got worse. Meaning, if you looked at features of patches and compared them through cosine distance to features of other patches in the image, at the 200K iteration mark, there was a very clear and distinct feature map. But as they continue training, at the 1 million mark, you see how it gets very noisy. So you no longer see these clear structures. So the gram anchoring objective, it's very simple and logical, but it's very cool that such a simple thing works, which is they ensured that the cosine distances of the different features across the patches remained consistent. And even at the 1 million mark, they wanted the cosine distances to look something like the 200K iteration mark. So using this objective, they were able to get not only global benchmarks and global metrics to improve, but also to get local metrics and local dense task performance to stay consistent. Which kind of also explains how it's able to generalize for various industry-specific or domain-specific use cases. Alrighty, yeah. So we spoke about the embeddings visualization for Dino versus Clip. Here it is in comparison in the same slide. You can kind of see what I was talking about here. So the intermixing of various classes is a lot more evident when you see them side by side. So for Dino, of course, you're able to see a lot more like specific broken out classes. And for Clip, you're able to see that it's more mixed in and kind of bleeds together. Another cool thing that FiftyOne has is the ability to not only look at embeddings at the full image level, but also look at embeddings at the patch level. So what that means is we're looking at crops of the images and we're cropping to the bounding boxes that are present in the images. So this kind of helps get rid of background noise and things like that. I looked at the patch level embeddings here. And a very quick note here, when I looked at this, I was like, this looks really familiar. What this looked like to me was actually a pest in and of itself. So I thought it was really interesting that the embeddings map looked very similar to a pest. So if you kind of squint really hard and quickly cycle between those two slides, you can kind of see what I'm saying. But yeah, I just thought that was a funny and interesting note to point out here. But with these patch level embeddings, right, you can also lasso different clusters and it'll return those patches or those bounding boxes that are of interest. And you can see that it does a really good job of clustering samples that have, or patches that have similar features together. So this is also another way to explore your data and then further curate it. Awesome. Okay, so having done all of this exploration and curation, the next thing that I did was train a model. So what I did to train this model was I broke out my data set into a train, val, and test split. So my train split was about 70% of these 19,000 samples, and then my val split was about 10%, and I left 20% aside for test. What I wanted to show you all was the histograms of distribution. So I think in Sinha's talk, somebody asked a question about what do the distributions between your test sets and your train and val test sets look like? Train and val sets look like. And that is an important thing to kind of look at to make sure your distribution of test is captured by your train and val distributions, right? So what you can do here is opening back up this dashboard. I can very quickly filter down to my training samples. So I tagged all of my samples that I wanted to use for training as train. So I'm able to filter that down. And you can see the distribution looks very similar. So the first class that's overrepresented in the base set, in the full data set, is also overrepresented in the training set. But I'm specifically interested, of course, in my test set to make sure that the test set distribution is well captured by my training set distribution. And that also, you can see, is very well captured. And yeah, cool thing about this dashboard in FiftyOne is these are all clickable bars. So for example, I can click on a bar here and it will filter down to those specific samples here. So I'm looking at my beat army worm. Let's see. yep, beta mealworm. I'm able to quickly look at the samples that contribute to this bar. So just further understanding and distribution validation here gave me confidence that the sampling method that I picked, which in this case was just random sampling, so nothing over the top. So the sampling method that I picked was able to capture the distributions and similar distributions in train, val, as well as test. But yeah, so I was able to trigger training directly from FiftyOne. So there's a fine-tune YOLO model plugin that we have. So I selected my detections field, which is ground truth, which is the field on which I want to train. And then I trained, I think, for 100 epochs, which should then show up in my runs tab here. So I think there's this model fine-tuner. You can see the hyperparameters with which I ran this. So trained it on the ground truth field and then trained for about 100 epochs. And then finally, what I did was once I got a trained model, I ran inference using that same trained model on my full data set. And then I was able to run model evaluation as well. So once I got this model trained, I applied that model on my full data set. So I applied this same model with the pest detection weights that I had fine-tuned, and then I got it to run on my full data set. And then I evaluated that model. So how evaluation looks like in FiftyOne is, let's open up the model evaluation tab here. I kind of kept the evaluation separate. So I wanted to evaluate on my test set separately and then evaluate on my training and val set separately, just to see, has my model overfit on my training and val sets when compared to my test set performance? So things like that. So just looking at test set performance here. So when I click into that model evaluation run, it automatically applies the tags for my test set, and it filters down to my test set samples, which is about 3,700, which is about 20% of the data set. So looks correct. We can see our precision was 0.7, our recall was also 0.7. Pretty okay precision and recall given that I was able to achieve all of this in 3 or 4 hours. MAP is the specific metric that I'm interested in because independent of my threshold, I want to see how good of a classifier the model is. And it looks like, I would say this is not that good, but for object detection tasks, this is pretty standard without doing too much curation. And then we can also start to look at class level performance, which is actually really cool. And here is where I want to kind of draw some insights, right? So I want to look at, are there failure modes that I see in my model? So let's look at this. So over here, right, in the thrips class, I see that precision is much lower than other classes. So let's look at thrips class performance here. We'll click on this thrips bar. It'll filter down to this, it'll filter down to the corresponding samples that fall under this thrips dataset or thrips class in my test dataset. And I think the first thing that stands out is definitely that it's underrepresented, right? There are only like 12 samples when compared to, let's click on another or different class here. Looks like there are 68 samples. So it looks like it is underrepresented in my test data set. So maybe that's a failure mode takeaway for me. A bunch of other takeaways are, let's also look at the same thrips in my test, in my training and validation data set to see if there's some incorrect labeling or something else that's happening. So it looks like ground truth for this one says it's a brown plant hopper, but my model thought this was thrips. But at the same time, this is in ground truth labeled as thrips, but this also kind of looks like thrips, right? And these two also look like thrips, but these are both in the ground truth. They're labeled as wheat, floio thrips. And they are, I think, lower class, which falls under the parent thrips class. So it looks like this, because I'm using the lower level taxonomy, I'm running into this like failure mode of, even though this is accurately classified, my model evaluation thinks it's incorrectly classified because I didn't have it roll up to a parent class. So I think my biggest takeaway from all of this is just that I should have used the parent level class. You can also look at the confusion matrix here and then click on various cells here, it'll apply the filter so I can further drill down and look at what's happening. So yeah, this was very quickly talking about the model evaluation. And yeah, so biggest takeaways throughout this whole process, I felt like using FiftyOne gave me a very quick way to iterate and come up with an initial solution. But I will say this is a good starting point and is not a deployable solution, right? Because there are very obvious failure modes also that I was able to surface from my investigation. And I think that also is a big value add in and of itself, that I was able to do failure mode analysis also so quickly using FiftyOne. So I'm going to, if I had more time to spend on this, the next thing I would do is use my takeaways about the distributions and things like that to feed back into another round of curation and another round of training and evaluation. Awesome. Thank you so much, everyone. I kind of breezed through the last little bit, but I did want to leave some time to answer questions.
Fantastic. The demo gods were very kind to you. Fantastic. Always
Yep.
very brave, very awesome. I think you did kind of briefly touch on this particular
Mhm.
point, but what was your methodology for how you split your data set into training, verification, and test? Did you do this randomly? Did you have some sort of method? What were you thinking there?
Yeah. We at Voxel 51 have a research team that's actually worked on core set selection and figuring out how to sample data accurately so that distributions are kind of maintained in the downstream like training sets as well as in the test sets. And Z-code is an algorithm that our company has released and our science team has worked on. But as part of that research, the other thing they pointed out was sometimes random sampling does really well. And for most use cases, random sampling does better than any complex heuristic that you can come up with. So I just stuck with random sampling and I feel like it did pretty well. Yeah.
So the next question is in regards to false positive and false negative images. Is 51 able to help you detect these types of images?
Yes, absolutely. So something that I didn't show you because I was kind of rushing through the last little bit is further filtering on the sidebar in FiftyOne. Let me actually very quickly show you since this came up. So going back to the data set here, once I have evaluations, right, I can actually look at false positives and true positives uh, at the field level. So looking at my custom YOLO test performance, I can look at, for example, true positives and false negatives. So let's look at true positives, for example, here, and I'm able to look at all of the samples that have true positives that were marked by my evaluator, and then false negatives. Let's get rid of two positives and just look at false negatives. I'm able to do that as well. So you'll see false negatives and true positives are labeled on the ground truth label field, and then on the predictions label field, you'll have the false positives labeled as well. So I'm able to also filter by that here. So I can add, oh, sorry, not best class, under YOLO predictions. I'm able to look at my false positives also. Yeah, you can also start doing pretty interesting things, Jimmy, like looking at your high confidence false positives to see where your model is overly confident for some reason and do more failure mode analysis there. Yeah.
Fantastic. And I think the last question, I'll kind of summarize it, which is going to be, what if your use case involves multimodal data? So not just images, but other types. How would you go about that in 51 and being able to perhaps train a model that can work with this type of data?
Yeah, great question. So with multimodal data, we have this notion of grouped data sets in FiftyOne. So you can group all of the modalities according to a specific timestamp or some other logic that you want, and you're able to look at all of your modalities in one place. So this is an AV example, but you're able to see I have LiDAR data as well as all of the camera modalities at the top. And a lot of agriculture use cases also uses LiDAR data along with cameras. So you can work with these types of data sets in FiftyOne as well, for sure. And that is something we've seen people in the agriculture space do.